Sasha Hafner here to talk a bit about software for measurement and prediction of methane potential. So what is this? This is free software for biogas research in a very broad sense of the word. You can work up lab measurements of biochemical methane potential, or BMP. You can do BMP prediction, and then you can do other stuff as well, like planning experiments. The software comes in two different forms. If you're familiar with the R language, then you want the biogas package, which is available on CRAN, and it's the more flexible option. If you are not familiar with R, then there's a web app version called OBA for the online biogas app. You can see that right here. So let's talk a bit about the three major types of tasks that you can carry out using this software. You can find more info on BMP data processing in an open access paper in the journal Software X. Both the biogas package and OBA use the same sequence of operations here. You start with your biogas measurements in the lab, and you work those up to calculate cumulative biogas and methane production and rates. And then the output, those data are combined with some information on what goes in each bottle to calculate BMP. So here's the web app OBA again, and it has three sections for carrying out different types of operations. We're going to work in the process biogas data section or tab here. So this example is done with some manometric measurements based on pressure, but there are multiple types of data that OBA can handle, and there's a lot of flexibility in how those measurements could be made. Over here on the right, you can see BMP results, in this case for an experiment that had 5 different conditions. There are a lot of different ways to calculate BMP, or let's say variations. In this case, the end duration was based on a relative rate, and we can see the relative rates here to decide when a trial should be ended. You can view and download your data through the web app, and there are a lot of different ways it can be displayed. So for example, you may be interested in methane yields for all times, and you can get them this way. I want to talk a little bit about a different type of data, very common in biogas research and in plant operation as well. So data from the AMPTS system. So I've used these with the application of what's called the LACIN protocol, which is one way of assessing the quality of your results. And here we see that all the criteria are met, and here are our BMP values. So moving on, let's talk a little bit about how you can predict biogas production from substrate composition and other optional information. So now we're going to work in the theoretical biogas tab or section, and here you can enter an empirical chemical formula, or if you have a pure chemical, then it's an actual chemical formula. And over at the right, now we have ethanol, for example. Over at the right, you see a maximum theoretical methane potential of above 700 milliliters per gram. So you can also look at the reaction and then get some additional details. If you don't know the exact elemental composition, but you have some nutritional analysis results, which are often uncertain, but you can still use them to make an estimate of the composition and then predict maximum methane production in that way. So you can see the empirical formula that was used right there. And if you select more details and then add some additional information, you can actually make an estimate of true methane production, not just the potential or the actual methane yield. So here we have to say something about the concentration of substrate VS, the substrate degradability, and the substrate partitioning. Concentration is used for carbon dioxide partitioning, which determines biogas composition or biogas quality. So this app will do CO2 partitioning. And you can see a difference, 56% versus 60% in the fraction of methane produced and the mole fraction that's actually present in dry biogas. So if you're interested in using this software or learning more, then you can join the mailing list by sending me an email message. You can check out the paper I mentioned, or you can just start using OBA, which is available at this URL, or the biogas package, which is available on CRAN and also GitHub.